Hi everyone. Today we are going to learn the NMJ, the Neuromuscular Junction, is a critical interface between the nervous and muscular systems, enabling the transmission of neural signals that ultimately result in skeletal muscle contraction. This specialized region is found at the point where a motor neuron from the somatic nervous system synapses with muscle fiber, forming a highly organized and intricate structure. The motor neuron originates in the anterior ventral gray matter of the spinal cord, with its cell body located within the gray matter's anterior horn. These somatic motor neurons are responsible for the voluntary control of skeletal muscle movement, receiving input from the central nervous system and then transmitting this signal down the length of the neuron axon. Upon reaching the muscle fiber, the motor neuron terminal forms a specialized synapse known as the neuromuscular junction, where the release of the neurotransmitter acetylcholine triggers a cascade of events leading to muscle contraction. In the neuromuscular junction, the motor neuron axon terminal closely opposed to the muscle fiber sarcolemma. The cell membrane of the muscle cell. Within this synaptic cleft, acetylcholine is released from synaptic vesicles in the motor neuron, diffuses across the narrow gap, and binds to nicotinic acetylcholine receptors located on the muscle fiber's surface. This binding even then initiates a depolarization of the muscle fiber's membrane potential, opening voltage gated sodium channels and propagating an action potential along the sarcolemma. This electrical signal is then transmitted deep into the muscle fiber through the transverse tubule system, ultimately reaching the contractile apparatus of the sarcomeres and triggering the sliding filament mechanism that results in muscle contraction. So the neuromuscular junction therefore represents a crucial site of communication and signal transduction between the nervous and muscular systems, enabling the voluntary control of skeletal muscle function that is so essential for human locomotion, posture, and a wide range of physical activities. In the intricate realm of neurophysiology, the role of the alpha motor neuron stands as a pivotal role or pivotal player originating from the cerebral cortex. Impulses travel down the spinal cord, eventually reaching these specialized motor neurons. The alpha motor neuron, specifically, is the neuron responsible for directly supplying the skeletal muscles with the necessary signals for movement. The mechanism behind this process is a captivating interplay of electrical and chemical phenomena. As the impulses from the cerebral cortex stimulate the cell bodies of the alpha motor neurons, action potential are generated. Action potentials are generated. These action potentials are the result of a delicate balance of ion movement across the cell membrane. Sodium channels sensitive to voltage changes open up, allowing positively charged sodium ions to rush into the neuron. This influx of positive charges causes the interior of the cell to become increasingly positive, a process known as depolarization. The depolarizing wave then travels down the length of the axon, the long projection of the neuron as the action potential reaches the synaptic bulb, a remarkable event occurs. The positive charge triggers the opening of specialized calcium channels, allowing calcium ions to flow into the neuron. This influx of calcium ions is the crucial step that initiates the release of neurotransmitters, the chemical messenger that cross the synapse, the gap between the neuron and the target muscle fiber. The intricate dance of ion movements, action potentials, and neurotransmitter release is the foundation upon which the alpha motor neuron exerts its influence on the skeletal muscles, enabling the precise and coordinated movements that we so effortlessly execute in our daily lives. The regulation of ion channels is a crucial aspect of neurophysiology as it governs the intricate mechanism underlying action potentials and synaptic transmission. Voltage sensitive calcium channels, in particular, play a pivotal role in this process. These channels are normally in a closed state with inactivation and activation gates controlling their opening and closing. However, when the membrane potential reaches a specific threshold, typically around typically around plus 30 millivolts, the activation gate of these channels opens, allowing calcium ions to flow into the cell. This influx of calcium is critical even as it triggers the release of neurotransmitters from synaptic vesicles. The synaptic vesicle containing the neurotransmitter acetylcholine are produced within the cell body, specifically by the Golgi apparatus, and then transported down the axon via the anterograde transport. Importantly, the acetylcholine itself is not synthesized in the cell body. Rather, it is composed of two constituents, choline and acetyl group, which are assembled at the synaptic bulb. The presence of mitochondria in the synaptic region also plays a vital role as they provide the necessary energy for the various processes involved in neurotransmitter release and recycling. This interplay between ion channels, synaptic vesicles, mitochondria is essential for the efficient and precise communication between neurons, enabling the complex functions of the nervous system. Understanding the mechanism governing voltage sensitive calcium channels and their role in synaptic transmission, a fundamental aspect of neuroscience with important implications for the study of neurological disorders and development of targeted therapeutic interventions. Acetylcholine is a crucial neurotransmitter in the human body, composed of two key components, choline and acetate. Choline is an essential nutrient is primarily obtained from the diet, as it is present in various food sources such as eggs, meat, certain vegetables. This choline is then absorbed through gastrointestinal tract, transported through the bloodstream, eventually crossing the blood-brain barrier and entering the synaptic bulbs of neurons. The other component, acetate, is derived from the mitochondria. The powerhouses of the cell are the sites of the Krebs cycle, a series of chemical reactions that generate energy for the cell. Acetyl CoA, intermediate in the Krebs cycle, is a source of the acetate required for the formation of acetylcholine. The synthesis of acetylcholine occurs when the choline and acetate molecules are combined through the action of the enzyme choline acetyl transferase. This enzyme catalyzes the reaction, resulting in the release of coenzyme A and the production of the neurotransmitter acetylcholine. The presence of this enzyme is essential for continuous generation of acetylcholine, which plays a vital role in various physiological processes such as muscle contraction, cognitive function, and autonomic nervous system regulation. Understanding the relationship between choline, acetate, and the enzymes involved in the synthesis of acetylcholine provides valuable insights into the mechanisms underlying the proper functioning of the nervous system. This knowledge can inform the development of therapeutic interventions in acetylcholine related disorders. So the regulation of neurotransmitter release at the synaptic cleft is a highly intricate and energy-dependent process that involves the interplay of various ion concentration and specialized proteins. The core of the mechanism is the proton H plus gradient established across the synaptic vesicle membrane. Typically, the proton concentration within the synaptic vesicle is higher than the proton concentration in the extracellular space. This concentration gradient is actively maintained by the direct utilization of ATP, process known as a primary active transport. By expending energy to pump protons against the concentration gradient, the cell creates high proton concentration inside the vesicle, which is crucial for the subsequent steps of neurotransmitter uptake and release. The established proton gradient serves a dual purpose. Firstly, it provides the driving force for the secondary active transport of the neurotransmitter, in this case, acetylcholine, into the synaptic vesicle. As the proton move down their concentration gradient from the vesicle interior to the extracellular space, they facilitate the co-transport of acetylcholine, thereby loading the vesicles with the neurotransmitter. Secondly, the proton gradient creates an electrochemical potential difference across the vesicle membrane, which can be harnessed to power the subsequent release of the neurotransmitter. This process involves the coordinated action of specialized proteins known as SNARES. This full form is soluble N ethylmalamide sensitive factor attachment protein receptor, which facilitate the docking and fusion of the synaptic vesicle with the presynaptic membrane. The SNRE protein involved in this process include synaptotagmine, a calcium sensing protein on the vesicle membrane, and SNAP25 and syntaxin, which are located on the presynaptic membrane. Upon the influx of calcium ions into the presynaptic terminal, synaptotagmine undergoes a conformational change, triggering the formation of SNRE complex and the subsequent fusion of the vesicle with the membrane, ultimately leading to the release of the neurotransmitter into the synaptic cleft. The intricate sequence of events driven by the proton gradient and the coordinated action of the specialized proteins ensures the efficient and regulated release of neurotransmitter at the synaptic junction and enabling effective communication between neurons and the propagation of neural signals throughout the nervous system. Synaptic transmission is a highly orchestrated process that involves the intricate interplay of various proteins in the coordination of calcium signaling. At the heart of this process are two key proteins, SNAP25 and syntaxin. SNAP25 is a critical component of the SNARE, which facilitates the fusion of synaptic vesicle with the presynaptic membrane, and syntaxin is, along with SNAP25 and synaptobrevin, forms the ternary SNARE complex responsible for membrane fusion. And this highlight the crucial role of calcium. The role of calcium in the synaptic transmission process. When the threshold of synaptic bulb is reached, calcium influx acts as a crosslink between the VSNREs and the TSNREs. VSNREs are synaptobrevin and the TSNAREs are SNAP25 and syntaxin. So these calcium-mediated interaction triggers a series of conformational changes, causing the SNRE protein to intervene and pull the synaptic vesicle towards the presynaptic membrane. The subsequent fusion of the vesicle membrane with the presynaptic membrane exposes the contents of the vesicle, including neurotransmitter like acetylcholine. When the voltage-gated calcium channels open, it allows for the influx of calcium ions, which is the second step in this mechanism. The third step involves the transport of choline, the key component of acetylcholine from the diet through a choline transporter. These choline is then combined with acetate in the presence of enzyme choline acetyl transferase, forming acetylcholine. The fifth step focuses on the active transport of acetylcholine into the synaptic vesicles, which is driven by the proton gradient established by the proton pump. The sixth step encompasses the interaction between the VSNRs on the vesicle membrane and the TSNRs on the presynaptic membrane. Their interaction triggered by the influx of calcium ions leads to the fusion of the vesicle membrane with the presynaptic membrane, enabling the release of acetylcholine into the synaptic cleft through the process of exocytosis. Lastly, the resting potential of the neuron is maintained by the activation of potassium channels, which allow for the efflux of potassium ions, rendering the intracellular environment for more electronegative. This delicate balance between the influx of calcium and the efflux of potassium is crucial for the proper functioning of the neurotransmission process, ensuring the efficient and regulated communication between neurons. The intricate process of neuronal signaling involves a delicate balance of ion movement across the cell membrane. When the resting potential is disrupted, potassium ions begin to flow out of the cell, driven by their equilibrium potential as described by the Nernst equation. This outward movement of potassium leads to gradual repolarization of the membrane, reaching a point known as the refractory period. And during this refractory period, the potassium channels distribute along the synaptic cleft, also affected, causing the membrane potential to reach a negative value of approximately minus 70 millivolts. And this drop is potential triggers the inactivation of the voltage-gated calcium channels, preventing the influx of calcium ions. Without the influx of calcium, the release of acetylcholine from the presynaptic terminal is inhibited, as the synaptic proteins responsible for this neurotransmitter release are unable to properly link together. The resting period thus represents a crucial stage in the neuronal signaling process, where the outward flow of potassium ions, the subsequent repolarization of the membrane, and the inactivation of calcium channels all work in concert to temporarily shut off the release of acetylcholine, and this temporary cessation of neurotransmitter release allows the system to reset, preparing it for the next cycle of neuronal communication.